Good morning. Welcome to Clubhouse.
Hi, thank you.
If you can click on my image and then click make moderator, then I can help in setting up the room. And nice to meet you. I'm Victoria.
Okay. Okay. I didn't know what I was doing clearly.
That's all good. Thank you. Well done. Welcome up, Jamie. And can you please tell me how to pronounce your name correctly?
It's Cecilia
Okay.
Lindestam Arlehamn.
Okay, nice to meet you, Cecilia.
You too, Victoria.
Hello, Doctor. Hello, everybody. How are you today?
Good, thank you. How are you?
Very well, thank you. Thank you for coming.
I sent the slides to Katarina, but I don't know if you guys have them.
She should be popping around in a moment. Yeah, and she'll take care of that.
Okay, great.
Yeah, normally we open the rooms for the researchers so that you have to do as little as possible.
Yeah, save
Yeah,
the
so
others trouble.
yeah, so
Yeah.
she'll be coming along, so you shouldn't have to do any other technical work except just enjoy the room
Yeah.
and enjoy delivering your research.
Okay, great.
Yeah, and then having some. There'll be a bit of Q&A. Oh, here we have Katarina.
Hi.
Good morning.
Good morning. Thank you so much for starting the room. How is everyone?
Very well, Katerina. How are you?
Good, good.
Yeah, good, thank you.
Good. Cecilia, where are you in the world?
Right now I'm in Sweden, but I'm working in La Jolla in California.
Okay, a little different. Yeah, well, Sweden is so beautiful, and I hope you're enjoying your time there.
Yeah, definitely. Where are you in the
I'm
world?
in Oregon.
Okay, that's also beautiful.
It is, and Oregon reminds me a lot of the way it looks. Reminds me a lot of Sweden.
Yeah, when I was there, I had the same feeling. I visited Portland, and it felt very Scandinavian.
Right, the evergreens and yeah, the plants and the sort of smaller hills.
Yeah, definitely.
And what has you in Sweden? Are you there for work or are you relaxing there?
I'm working from here, but I'm here for my father, who actually has Parkinson, so needed a bit more help. So I'm sort of spending time both here and in California.
I'm sure he appreciates that very much.
No, he does. Yeah, for sure.
Yeah.
That's an amazing connection. I know that I have at least one friend who's coming today because one of her parents also suffers from Parkinson's disease. And I was thinking about your research, that there's research that's shared here that's so fascinating, and and we're just driven by curiosity. And then there's also research that people are waiting on desperately,
Yeah,
because of
definitely.
human need, and that's yours.
Yeah, there's a lot of people affected by Parkinson's.
Is this your first time in Clubhouse right now?
Yeah, it is. Well, except for the initial, we had a little pre-meeting
Okay.
that Katarina helped me with. Clearly, I didn't learn
Oh,
enough.
you were great. I mean, look at what you did. You opened the room by yourself. Clearly, you learned a lot.
No, definitely.
I
Yeah.
was like, I just want to be on time.
Yeah, that's
Then
great.
it said start room. I'm like, I guess so.
I know it's a little tricky because that opportunity is there, and it's so hard to ignore. I was asking because I hope that you will, after here, if you have the time, that you might consider to look around and see what else is
Yeah, I
happening
should
here.
definitely do that. I'm
Mhm.
sure there's more of these types of talks and things. So
Well, there are all kinds of topics. We try to create here in Science Society a welcoming and inclusive space that researchers can feel comfortable presenting in and know that their work will be respected here, and also for listeners of all levels of science knowledge to feel comfortable and know that they can come here and listen, and even put questions in the chat. Sometimes people will put questions in the room chat or just ideas that they're thinking of, so there can be a lot of audience engagement.
sounds great.
And this is really
Yeah.
just a place.
Oh, go ahead.
No,
No,
please,
I
Kevin,
just
you go
wanted
ahead.
to say welcome everyone, and we have 2 more minutes and then we'll start. But go ahead, Jamie.
Oh, no, I was just going to say to Cecilia that this is quite a relaxed place. Like, we're all pretty curious and we like to keep things, we really like to get our teeth into the nice juicy science, and but we're also quite relaxed. So if you just take off your shoes, don't worry about your hair.
I know it's not on video, so
Yep, you can just relax it and just enjoying explaining your work, which we're very, very excited to actually read about.
Yeah, and I'm happy to answer questions if anything is unclear as I go along, or if you want to wait till the end.
That is totally up to you. Different speakers prefer different things, so it's really, if you're comfortable having a two-way dialogue thing, um, we can accommodate that. Or if you would like to speak for a
Yeah,
while,
definitely. Sometimes it's just easier if you think of a question to ask it straight away in case, um, or if you want to hold on to them. I'm fine either way.
we'll try and do our best to not interrupt your flow, because quite frequently, if we jump in too quickly, we're about to ask a question you were about to answer anyway. So
Yeah, then I'll just tell you.
exactly. Oh dear.
Okay, I think we can slowly start. Thank you so much, Cecilia, for starting the room and for everyone to be here. Thank you. It was amazing. Yeah, welcome everyone to the Science Society. We have our special guest speaker here today, Dr. Cecilia Arlehamn. And let me give you a little bit of background information first, and then Victoria will ask a more general question, and then the stage is yours for your presentation.
Okay.
So, perfect. And Cecilia, Dr. Arlehamn, she's a research assistant professor at the Center for Infectious Disease and Vaccine Research at the Loyola Institute for Immunology in California. And she did her master's degree in 2005 in biomedicine at the Calmer University. And at the University of Glasgow, she worked in molecular functions and disease until 2006. And then she did her PhD in microbiology and immunology at the University of Glasgow in 2009. And now she has her own lab, and it is focused on understanding T-cell immune responses to Mycobacterium, tuberculosis, and other infections, as well as self-antigens in the context of neurodegenerative diseases. So yeah, she's recently also focusing her research on Parkinson and Alzheimer's disease. And this work has identified an interesting target for T-cell immune responses, and that's what we are talking about today. So yeah, welcome. Thank you so much for taking the time. And Victoria, please go ahead.
Yeah, thank you, Katarina. And as Katarina said, Science Society welcomes you, Dr. Arlehamn. We're really excited to hear you discuss your work in Parkinson's disease-specific gene signatures and learning from you. And so to bring another human aspect into the presentation this morning, well, I'm on the West Coast, as you know now. I have a question about your connection to science. So if you look back through your life, is there a moment or experience that you associate with providing the spark or of curiosity that helped you develop your interest in science? You know, maybe a class or a book or,
Yeah,
you know,
definitely. I
thank
mean,
you.
and thank you for having me today. I wanted to be a vet uh, during school, but realized with my cat allergies that that's maybe not a good idea. So then I studied biology for a year before I started in University of Kalmar. And there I did a course in cell biology and just found that really fascinating and thought this is really um, what I should do. This is what I want to do. So that's where the science aspect came in. So then my undergrad, and then going on to PhD, and then postdoc, and now I have my own lab.
Yeah, thank you. That's an exciting path. And I'm glad to hear that you're working in beautiful areas. La Jolla is also really lovely. So that's important. I think it's inspiring. So yeah,
True.
so here you are. And then follow-up question is, how does this work bring you to the research that you're working on now? What was your path there?
So I did initially, when I joined the lab in La Jolla, I worked on Mycobacterium tuberculosis. But the techniques can really be applied to other diseases as well. And we were curious whether there's a T cell component in Parkinson's. And that's what really led to what I'm going to talk to you guys about today. We discovered alpha-synuclein specific T cells in Parkinson's disease, and that's really taken off a new area of research and a lot of work, and a lot of questions that we still want to answer. So that's a big focus of my lab.
Thank you. Thank you very much. I think it helps us to, just to imagine the person who is doing this research as we hear, as we hear what you're sharing. So to continue, you have the floor and you may launch into your presentation. And then, as Jamie had mentioned, whichever format you'd like. It's more traditional to wait until the end of your presentation, and then you can invite questions as you'd like. There have been some presenters who have said, can you please feed us some questions because it will help us flesh out the body of our talk. But
Yeah, I mean,
which
like I
is
said, if anything is unclear or if you have questions, I'm happy to answer them as we go along here. So no
all
problem.
right. Well, thank you. Well, the mic is yours, Dr.
So
Arlehamn.
thank you. So I hope everybody can see the slides. I thought I'll talk about our recent work defining Parkinson's disease specific gene signatures. So I'm at the La Jolla Institute, like was said, and then at the bottom left side, you see the funding agency that funds a lot of our Parkinson work. So if we move on to slide 2, I just thought I'll give a little introduction to what Parkinson's disease is. Just so we sort of set the stage for the talk today. So it's a degenerative disease that affects nerve cells in the brain, and particularly nerve cells that are in the substantia nigra of the brain. So you could see that on the picture here. So in Parkinson's disease, these nerve cells die. So you get less dopamine, which is an issue. That's when you start to experience these symptoms that are traditionally thought of as Parkinson's disease, with the movement disorders and so on. And actually, worldwide, there's more than 6 million people that have Parkinson's disease, and there's 1 million in the US. And with the aging population, and as we live longer, this will only increase and is projected to increase. So there's a huge need for different therapies and diagnosis and treatments for Parkinson's disease. So if we go to the slide, that's an iceberg. Really, the motor symptoms is just the tip of the iceberg. So there's a lot of what you traditionally would see as Parkinson's, like a forward tilt, reduced arm swinging, obviously the tremor that most individuals will experience of the extremities and the head potentially, and then slurred speech. But there's a whole range of other symptoms that also occurs in Parkinson's disease, and it's not something that you would necessarily see, so such as anxiety, depression, constipation, cognitive issues, sleep disturbances, and a whole range of other issues. So that's not just motor symptoms, but a whole range of other symptoms. So if we move to slide 4, there is no test that can say, yes, you have Parkinson's disease. There's a lot of different aspects that a clinician will use to diagnose you, but there's no specific test. And there's also no marker how quickly you're gonna progress in your disease. So that's something that's needed, of course. And importantly, there's also no cure. So the medication that you get can only help with managing symptoms. It can't cure your disease, which is obviously a problem as you go down that path. So on slide 5, I just want to mention a little bit what we do in the lab and our focus, which has really been inflammation in the broad sense, which if we hear inflammation, we tend to think of that as a bad thing, right? It's the cause of many diseases. It can cause tissue damage. It can linger. But actually, inflammation is the immune system in action. So it is normal, and it is important because, of course, we want our immune system to react to whatever pathogens we experience, or other things that we end up through our lifetime. So on slide 6, the goal is, and that's the motto of the La Jolla Institute for Immunology, is life without disease. And there you really want a balance between immune activation and immune suppression. If you have too much immune suppression, you can have these, for example, cancer or chronic infections. And if you have too much immune activation, you can end up with autoimmunity and allergy. So that the goal is a balanced scale, but obviously your experiences and everything also contributes to that. So if we go to slide 7 next, a hallmark of Parkinson's disease is really this accumulation of aggregated alpha-synuclein in the brain. So here is a picture of the substantia nigra, where you can see these dots, which are meant to represent aggregated alpha-synuclein, which is seen in Parkinson's disease. And these aggregates are called Lewy bodies, and they can be taken up by particularly a type of cells in the brain called microglial cells. So that's sort of a hallmark, and we started off by looking to see if there's a T cell component of the disease, so sort of an adaptive immune response involved, which could point to an autoimmune feature of Parkinson's disease, which hadn't been shown before. So that work led, if we look at slide 8, to a publication in Nature, where we found that T cells from patients with Parkinson's disease recognized alpha-synuclein and T cell epitopes, as they're called, or these peptide sequences that activates T cells. And we see more of those in Parkinson's disease individuals compared to healthy controls. So that was very exciting. And as I said, it led to a lot of additional questions. So on slide 9, there is a potential, and we don't know how this works yet, right? But if the microglial cells present alpha-synuclein and activate T cells, they could potentially contribute to the death of the neurons in the substantia nigra and contribute to the progression of the disease. So on slide 10, I've just sort of made a progression between slide 10 to 12. But if we start on slide 10, the development of Parkinson's is a very long and gradual progress. And if we start off here in the sort of light yellow, you have all of your substantia nigra neurons remaining, and you're in sort of a risk phase. And then as the neurodegeneration starts to happen, you don't have a lot of symptoms, but you're in a sort of preclinical phase, and there's no tests to determine this, so that's not something someone would know. And if we look at slide 11, you then progress and you start having non-motor symptoms. Some individuals report constipation and loss of smell, but obviously that happens for many other diseases, so it's not specific, of course, to Parkinson's disease. And then you start having slight motor signs, and you can see as this goes on, the number of substantia nigra neurons slowly decrease over time. And obviously this, as it's called, prodromal phase, can last for more than 10 years. And then if we go to slide 12, once you're diagnosed with Parkinson's disease and you have your motor symptoms, which is usually what makes you see the neurologist in the first place, unfortunately, most of your substantia nigra neurons are already gone, and those won't come back, so to speak. So really what's of huge interest is to find a biomarker for early detection, and then ways to delay or prevent this neuronal degradation, because that's really what would help individuals with this disease. So if we move to slide 13, so one of our goals in the lab is to understand the prodromal phase of disease and develop biomarkers to detect these. And our question is really if T cell or T cell reactivity could be useful as a potential biomarker of disease, and even a prodrome marker, so that you can see it before you start having these clinical symptoms. So on slide 14, It's the same picture again, but just to say that we looked at longitudinal changes in alpha-synuclein specific T cells. Since this disease takes, fortunately, one could say it is slow progressing, but it's very hard to follow longitudinal samples. But we were fortunate to be contacted by someone who, for other reasons, had white blood cells cryopreserved, and we could look at the reactivity in their samples. And we found that in this prodromal phase, you see higher reactivity in your T cells against alpha-synuclein than you do in the clinical phase, so after they were diagnosed with Parkinson's disease. So that's very interested, and we're now interested obviously in looking at more of these types of samples. But if we look at slide 15, we also looked at individuals that were already diagnosed with Parkinson's disease, and they had different times from diagnosis. So someone could be diagnosed very recently, so within a year or so, or someone could be up to almost 30 years since their diagnosis. And then we measured the immune response to alpha-synuclein in these individuals. And you can see in the graph here that a higher immune response, so each dot in this graph is an individual, and the higher immune response is found in more individuals when they're closer to diagnosis compared to when they're further out. And we were able to replicate this in independent cohorts. Of course, that's something you want to do as a researcher to make sure it's not something odd going on with a particular cohort. And what was also interesting, some of these MoCA and UPDRS are some clinical scores really that is used to diagnose Parkinson and also look at the progression of the disease. But we didn't see any correlation with reactivity compared to those. It was really the year since diagnosis that played the most, that was correlated with the immune response. And linking that to the individual who we could measure before diagnosis, it seems like we have higher immune response before onset, and then that sort of trails off as you progress. So if we go on to slide 16, we think that in the lab we have a model to study Parkinson's disease, just to look at peripheral T cells. And this work that I just described to you was published not that long ago in Nature Communications. So moving on to slide 17, where it's really the bulk of today's talk, where we were curious to see if we can use transcriptomics or gene signatures, whether that could help us identify these diagnostic or perhaps mechanistic markers of Parkinson's disease. And with the sort of thought that alpha-synuclein specific T cells, if you have those, that's a sign of inflammation and ongoing autoimmunity. And individuals that don't have that may have had it in the past. So we're just taking a snapshot at one time point. So one of our other interests is really to look at even longitudinal samples for individuals with Parkinson's disease. But you see the same graph as I showed you a couple of slides ago, where we divided our cohort into those who respond to alpha-synuclein, so those are called PDR, and those that don't respond, and those are called PDNR for non-responders. And this has been published this year, so I just put the reference here if you want to look at that. So looking at slide 18, that will explain what we did. So we did RNA-seq just by taking PBMCs, so those are the white blood cells from a person, and here I've just shown flow cytometry plots how we sorted those cells. So we take a sample, run them through a machine, and get them as a single cell suspension at the end that we can look at their RNA expression. And then we also looked at CD4 and CD8 memory T cells, so just these two most prevalent types of T cells in the blood. And we focused on memory cells for those populations based on markers called CD45RA and CCR7. And that's shown here in this diagram. So if we look at slide 19, we actually do these comparisons, right, to find differentially expressed genes. So genes that are different between these samples. And our initial look at this, we compared all the different cell types that we had. So that's shown here in what's called A on the slide, and that's a principal component analysis. And you can see here that if you have healthy controls or Parkinson disease individuals for these different cell types, so CD4 in sort of red pink color, and CD8 is in green and light green, and PBMC is in blue, you can see that they cluster based on cell type, and they don't necessarily cluster based on whether it's a Parkinson individual or a healthy control. And if we look at the overlap in B, you can see there's very few, first of all, few genes that are differentially expressed, and there's very little overlap between the cell types. So that was looking at Parkinson disease individuals as a whole. But if we turn to slide 20 instead, we can see that when we classify the subjects based on their response to alpha-synuclein, so like I said, the responders versus non-responders, and potentially then someone with autoimmune features compared to those that do not have that. If we look at slide 21, you can see that the number of genes goes up. So we find a lot more differences when we take that into consideration. So the first three rows just shows Parkinson disease individuals as a whole versus healthy control non-responders. And then we look at Parkinson responders versus non-responders, both for Parkinson and healthy controls. And you can see we have a lot of differentially expressed genes. And if we dive into those signatures further and look at slide 22, this is a busy slide. Here we're just looking at the differentially expressed genes. So if we, for example, focus on panel A, you first have a volcano plot where you can see how the genes are expressed and how different they are between the responders versus non-responders. And then when we take that gene signature and look if we can cluster the different groups of and the different cohorts, you can see that the Parkinson responders forms one cluster, they're in red, whereas the non-responders forms a different cluster. And this is true for PBMCs, CD4 memory T cells, and CD8 memory T cells. So we have a signature for all the different cell types that we studied. And interestingly, some of these differentially expressed genes are genes previously shown to be involved in Parkinson's disease, and not necessarily in T cells, mainly neurons, but we find differential expression of those. So if we go to slide 23, we perform something that's called a gene set enrichment analysis, where you can look at the genes, the group of genes that you have identified, to see how are they linked together, and what are they shown in the past to be involved in. And in this example, we're looking at an example of genes that were involved in Parkinson's disease. And because you have a peak closer to the responders, so in red, the PDR, that shows that there's an enrichment for this Parkinson disease gene signature in those individuals that respond to alpha-synuclein compared to those that do not. And it's just another way of showing you that those genes that were previously linked to Parkinson's disease in neurons, as I said, also show up in this case in the CD4 memory cells. And if we move on to slide 24, we also looked at not only the gene expression, but the protein expression. And here I'm just showing some of the examples in, for example, PBMCs, where we have the lowest expression of CCR5 in Parkinson responders, and then it sort of increases as you go through Parkinson non-responders and healthy control non-responders, and to the right there under PBMC you see the protein expression. And so it ties to the protein expression essentially. And now we're working on, if we go to slide 25, trying to look at these genes in more detail. So look at the top differentially expressed genes and then see where they are expressed. Are they membrane bound? Are they secreted from the cell? Are they intercellular? And then also cell specific responses or expression that we can see to see if they're a potential target that we could use and develop antibodies against, or use antibodies that are already available and see how that modulates the T cell response against different antigens. So on slide 26, some take-home messages. That we see that alpha-synuclein specific T cells are most abundant before diagnosis of motor PD. And I haven't shown you this, but well, I kind of have. You saw on the graph that not all Parkinson individuals respond to alpha-synuclein. So do they respond to a different neuro-derived antigen? That's something we're looking into right now, and also looking, as I said, at these longitudinal samples. We find a gene signature in these alpha-synuclein specific T cells, and the gene signature is enriched in the Parkinson's disease signature. And we also shown that by classifying the patients into responder versus non-responder, we can identify additional granularity and information that might be important for these individuals. And with that, we're on slide 27. And obviously we don't do any mouse work in the lab, so we really rely on this generous participation of patients that sign up for our study, and we're very grateful to them for their donations. And listen here the different sites that we're working with in this project. So very focused, it seems, on the East Coast and West Coast, but that's how that ended up. And on slide 28, we look at this autoimmunity and the role of T cells with the goal of improving diagnosis, understand the progression of the disease, unlock this key so that you can interfere perhaps before all the neurons are already gone, and then find gene signatures that contribute to the immune response, with the goal, of course, of stopping Parkinson's. And then the last slide is just acknowledgements of everybody involved and also funding agencies. So, and thank you so much for coming to this talk today. I'm happy to answer questions. I realize I don't have no idea how this platform works. So if you try to say something and I missed it, I apologize.
Oh no, thank you so much. This was great. I really appreciate it. I wanted to ask, and then if anyone has questions, please flash your mics. My question was, so do you think that because you have this T cell response so close to or pre-diagnosis, what do you think, what role does the immune system play to maybe also make the disease worse, or do you just think this is just purely a response and doesn't contribute in any way to the disease
No,
progression?
so right now we think, based on other evidence in the field, that it seems that the T cells perhaps have a detrimental role. So they respond and then they contribute to the killing of the neurons, which would obviously make the disease worse. There's not a definitive proof for that though, so that theory may well change, but that's the sort of ongoing hypothesis.
And so if that's the case, is there maybe a difference in people with Parkinson's in their T cells, that they are kind of triggered more easily, or over responsive, overactive?
So studies have shown that individuals with Parkinson have higher levels of certain inflammatory markers. What we tried to do, since we're a T cell lab, and that's actually out as a preprint right now on bioarchives, but we wanted to see if that extends to T cell responses against commonly encountered antigens. And by that I mean coronaviruses. Obviously, they're a hot topic right now, but there's a lot of what's called common cold coronaviruses, so not necessarily what we've been experiencing for the past 2 years. And then also looking at other viruses, flu viruses, rhinoviruses, which also causes colds, antigens that are found in vaccinations like pertussis and tetanus, and sort of chronic viruses like CMV and EBV. But we don't see a difference. So the hyperinflammatory response in PD individuals doesn't, at least against those antigens, translate to higher reactivity there. Instead, it's similar to age-matched healthy controls, and I want to point out that it's obviously important to age match, as a lot of individuals with Parkinson's are it's most frequent in individuals over the age of 60. So you have to compare to a similar cohort of individuals without Parkinson's. So it doesn't translate there, and that could be, obviously we see higher responses to alpha-synuclein and potentially to other neuroantigens. So perhaps it's more specific than just higher reactivity to anything.
Interesting, thank you. Yeah, please go ahead. Flash your mic if you have a question. And yeah, Dr. Shark, go ahead.
Yeah, thank you so much for wonderful work, and you shared that with us. My question is about that previous exposure that you just mentioned, and I know that you had a research around the TB. I
Mm-hmm.
mean, I was just wondering, did you find any significance between the exposure with the TB or other kind of viral exposure and your finding? This is my first question.
No, no, we haven't. So, and for TB, I haven't looked in at that in detail. Of course, in the US, where most of our samples come from, we're very fortunate to have very little Mycobacterium tuberculosis infections. So that would require quite a large cohort to look at that, whether there's anything, and I don't think that's been reported. Um, no, so we haven't found anything in the T cell response linked to previous exposure that's higher in Parkinson compared to controls.
Yeah, I mean, your result totally makes sense for me in practical level and about the neuroinflammation that you just mentioned because too many of the patients, I mean, report the same symptoms. I
Mm.
mean, also we are seeing the IgG and IgM positive in those patients. And another question is about different types of neurons because we know that about dopaminergic neurons, and we have
Mm-hmm.
10 types of the dopaminergic neurons. So your report is matched with all of them or one of them? Because one of them I know specifically it's very important
you
and it's
you
related
um
with
you
53.
seem to be a lot more knowledgeable in this area than I am, I have to admit. So um we haven't I want to point out we haven't specifically looked at neurons in our group. What we look at is peripheral T cells that are in the blood that respond to this protein that is found aggregated in neurons in the substantia nigra of Parkinson individuals. So we don't have any data on specific neurons and which ones are affected more than others. So I would have to refer to other people's work for that, and I'm afraid I can't answer that question.
Thank you, Cecilia. That was wonderful.
There are a few questions in the chat. So one that I wanted to read out was Girish question. Have you looked at glycan age signature in patient samples? Glycan age equals glycosylation of circulating IgG bodies that increase with age. It is a robust marker of long-term chronic inflammation.
No, we haven't at all looked at any antibodies. No.
Okay. He's adding, it's akin to the HbA1c for glucose correlated with a number of age-related disease, including Parkinson. I wonder if it can help with early preclinical diagnosis when coupled with T-cell genetic signature and other biomarkers.
Oh, I see. Yeah, I don't know enough about this, but it sounds like something that could potentially be measured. So I thank you, Girish. I should go and read up on that and see if that's something that we should look at in our patients.
If you don't mind, I can answer for the patient that they have a higher A1C or even they have I mean, they are on the age of A1C, we do prevention. I mean, because one of the result is the neuroinflammation, so it's correct.
Thank you.
Does anyone else have a question? I saw that LT came to the stage. Here.
Yes, great presentation. Thank you. My question is, since you're looking at this inflammation in the brain, do you also find, and of course you're looking at the Parkinson, is there like I know a family, the grandma has Parkinson, of course, in her 60s. The
Mm-hmm.
mom is okay, but that daughter, so we're talking about the second generation, so that daughter, my generation, has
Mm-hmm.
Parkinson. So in her 30s, in your family, I mean, in the cohort you study, do you see a pattern, like skip a generation? That's my question.
Um,
Thank you.
Thank you. No, we don't really look at that. So the individuals that we have seen so far um, just donate once, and we ask them for their history in terms of also family history of Parkinson's disease. But we haven't looked at that in detail, and we also don't have enough patients to draw any conclusions. To do those types of studies, you need way more data than what we have blood samples from. What I can say is for the healthy controls that we use, we also ask them for their history in their family, and if they say sort of like you're describing, my grandmother had Parkinson, they're not they can't participate as a control, since that might confound things. But yeah, I hope that somewhat answers your question.
Yes. If I may, can I ask a related question? Is there a, you don't get the human, right? Do you know like a good animal model then? That's my follow-up. Then I'm done with question. Thank you very much.
So there are mouse models where you can induce Parkinson-like symptoms and through viral vectors and this sort of thing. But they don't, right? They don't develop Parkinson's by themselves. You need to induce those symptoms in them. Um, I think I don't know whether there are sort of non-human primates or that type of thing. Obviously, um, it would be very expensive since it's more prone in individuals that are older. So and also if you don't have a particular reason to look in primates, you wouldn't necessarily do that. So there are mouse models, but in terms of whether you can skip a generation, I don't think that's, you can't do that in mouse models. You would have to look at large cohorts of people.
Thank you very much.
I don't know, Leslie, Eli, did you have a question? Just checking in. If not,
This is just clarification on a point. You mentioned at one point that there was enrichment of the Parkinson's disease gene signature in the T cells. Was that specifically for T cells that respond to alpha-synuclein or T cells in general?
So that's a good question. Thank you. No, so we looked at T cells in general, right? We didn't divide them into alpha-synuclein responding versus not. So of course, not all the T cells in a person will respond to alpha-synuclein. So I don't know. What I can say is it's in an individual that have T cells that respond to alpha-synuclein, but we don't know within the T cell population what that looks like.
Yeah, that
And
the,
that's
the
something we would like to do, to sort of do single cell sequencing and look at that in particular, but we don't have any data for that right now.
Yeah, because it that raises the question of whether there's something affecting the T cells that make them more prone to respond to alpha-synuclein, and this is affecting all T cells versus something that happens to the cells that are responding to alpha-synuclein. And the things that came to mind were things like exosomes.
Yeah, I've heard a lot about exosomes actually recently, not particular for this, but also for tuberculosis. So I think that's certainly up and coming in terms of things that people are looking at.
Yeah.
So, oh,
No, no, go
I
ahead.
had a,
Go ahead.
yeah, I just had a quick question. Since Parkinson's disease is more a collection of symptoms and seems to have a lot of different causes. It was initially classified as an environmental disease, or was discovered as one. Do you see any variability in the T cell activity that you're measuring between different Parkinson's disease patients, as far as perhaps classifying what the well, don't want to speculate as far as classifying different causes, but just is there variability?
We don't see any right now, but also, like I said before, those types of studies require a lot of patience, and our cohorts, you know, they're certainly large for individuals that donate cells and blood for research, but they're not that large. So we can't really draw any conclusions. What we're starting to look at now is certain individuals. There are some genes that are identified as risks for Parkinson's disease, so certain mutations, and we're starting. Those have not been included in our studies because it's you know, another factor to take into consideration, but we're starting to look at individuals that carry those types of genes to see if there's differences in their T cell responses. But I not so far. We haven't found anything so far.
What about a different tack for possibly finding prodromal people? And we have a large cohort of people with sleep disturbances,
Yeah,
IRB, in
and
Minnesota, that I'm in looking at movement disorders and in general, and in Parkinson's disease, and other people in my lab are working with the possible prodromal. I mean, so many people go on to get a Parkinson's-like disease from that. It's within 10 years, so
yeah.
about
No,
two-thirds.
thank you. That's really important, and obviously I skipped over that in my talk, but that's another thing we are looking at. So recruiting individuals with RBD, exactly for the reasons like you say, that unfortunately a lot of them go on to develop Parkinson's disease, and we're trying to follow these patients longitudinally to measure whether they have higher reactivity to alpha-synuclein compared to controls, and or other antigens that we're looking at. So yeah, certainly that's a huge interest and something we're actively working on to look at.
Okay, good. Just wanted to comment, it's a bit of a chicken and an egg thing at this point, because there's a lot of research efforts focused on identifying people at the prodromal stage, but we don't really have much in the way of mitigation to offer at this point. So,
Yeah, exactly. No, I completely agree. It is a chicken and an egg, and to some extent, you know, do you really want to know if you're gonna develop Parkinson or not if there's nothing that can be done about it, right? But we obviously need to do more research and try and find those things out so that they can be helped earlier.
well, if both sides have success, then we'll be golden. So, thank you very much for your talk.
Thank you for coming.
Um, please go ahead. I see Ben and King Yin Yan. You also came to the stage, Katie, David. Please go ahead.
Yeah, I was just curious. Do you see any further potential applications in the commercial clinical setting of this work in terms of whether developing a point of care diagnostics or perhaps in drug development to attempt to address the Parkinson's disease from various different angles, whether it's cure or a relief of symptoms or anything like that. Did you see the sort of commercialization prospect of this research?
No, I mean, in the sense what I mentioned, where we have potentially higher T cell reactivity, right, in the prodromal phase. So that could potentially be looked at as a biomarker. But there's obviously a lot of things still remaining to do. And in terms of the gene signatures, that's why we're looking at whether modulating some of these genes has an effect, and whether that, eventually could go into animal models and then, trials, and see if that has a positive effect. Obviously, you don't want to make anything worse. So, yes, there is a potential, but I think it's still some way away.
I mean, so just to follow up, as far as you know, are there any companies, whether biotech or pharma, that are looking at something like this or have been working on something like this? Or is it just completely virgin territory that nobody has really thought about doing something like this before?
No, so I think right now it's basic research. I don't know of any companies that are looking at this type of work that we've been describing.
Katie, David, do you have any questions?
Hi there, this is David Perez. I just kind of joined late. I wasn't exactly sure what you had discussed so far in reference to, it sounds like you were talking about some gene therapy, correct?
So I was discussing differentially expressed genes that we see in individuals with Parkinson that respond to alpha-synuclein compared to those that do not. So in terms of T cell responses, that was the focus of the talk today.
And your CD4, CD8, and what was the count after that therapy? Was it above 1.0, the ratio?
Um, so we certainly see an increased amount of differentially expressed genes in the T cells when we look at individuals that respond to those that do not. I'm not sure I understand your question.
So I have some experience. We did some studies with Alzheimer dementia patients for 3 years. It was a rolling 12-month admission. And we saw some very powerful, markers. TNF alpha was down by 50%. The BDNF was up by 11%, and the CD4, CD8. A lot of the markers that we found, like the CD14, which is the macrophages, would help remove some of the beta amyloid plaque. We saw some of those significant improvement over just with some dietary supplements, which was interesting. Basically, rice bran, aloe vera, and some IP6 and acetyl hexophosphate. So I'm seeing foods making a big difference in some of the gene expressions and some of the research that not only we have done, and they're peer reviewed and published in the Journal of Alzheimer's Disease, but also some of the work that Elizabeth Ryan has done in rice bran at the University of Colorado in Fort Collins. She's seen some very unique things happen. She's documented over 450 phytonutrients just in one molecule, rice bran, which are amino acids, minerals, cofactors, and we're seeing a lot of different things happening there. And I just spent a month in Budapest, and I saw some oxygenated water have over 600 genes it regulates, that it downregulates and upregulates, just oxygen water. So hypoxia is, I think, a good area to investigate when we're in a hypoxic state. Things are happening in the body that not only help people with Parkinson's, but also Alzheimer's and various forms of cancer. So that's an area that I've been, for the last 7 months, spending a lot of time on in the area of hypoxia, lack of oxygen to our cells. Well, most people think hypoxia is climbing a mountain, lack of oxygen to the brain, but hypoxia is the culprit for many things because it's caused, by
Do you have
stress.
I'm sorry to interrupt, but do you have a question in there? Because we are almost up to the hour and we were about to close the room. So if you could now ask your question, that would be nice.
No, I'm just learning, listening. Thank you.
Thank you. And David, also, you can listen to the replays so that even though you did come to the room late, you still can have an opportunity to hear everything that Dr. Arlehamn has shared with us. Thank you so much for joining us.
Okay, then let me read out 2 more questions in the last 2 minutes we have from the chat. So Feng Trang, he said he would love to see ED50 comparison data versus L-DOPA in NHP rat PD models. I'm not sure if you have a comment on that.
No, I don't know. No, that's very different from our work.
Okay.
So yeah, I don't have a comment regarding that.
And what is the Go classification pathway enrichment of PD signature and T cells from Girish? And sorry if you missed that.
So I didn't go into that in detail, but we do have all that data available in the manuscript. So if you look that up on MPJ Parkinson's disease, those analyses should be in there.
Yeah, if anyone needs maybe the papers as PDFs, please reach out to me. I can send them to you, share them with you through my Google Drive. And yeah, thank you so much, Cecilia, for taking the time to share your very interesting work with us here today. I really appreciate it, and I hope you come back one day. Give us
Yeah,
updates.
thank you. Thank you for having me, and thank you to everybody who took the time to sign on today. It was great sharing our research and hearing your thoughts as well and questions.
Yeah, thank you. And yeah, all the best for you and your research. It's very important. So thank you for doing it and investing your life into it. And
yeah,
yeah,
thank you.
Yeah, thank you very, very much for coming. Thank you.
Take care, everyone.
Thanks, Cecilia.
Thank you.
Thank
Bye,
you.
everybody.
Thanks.
bye, everyone. Yeah, and thanks everyone for coming and asking questions. And if you like this type of rooms with guest speakers talking about their work, please follow the Club Science Society. And tomorrow we have a room, the researcher Dr. Sarwat from IBM in Switzerland. He's coming to talk about neuromorphic computing and neural networks. He will share his latest 2 papers about that. Yeah, and we'll have more rooms next week and the following weeks. So yeah, thank you everyone. Thank you, Cecilia, again, and we'll close the room in 3, 2, 1.
Thank you.
Bye,
Thanks,
everyone.
Katerina.
Thank you, Katerina. Thank you, Doctor.
See you next time. Bye bye.
Bye.